Welcome back, everyone. Today, we're diving into a topic that's both fascinating and incredibly challenging. Glioblastoma.
Or GBM, as it's often called.
Exactly, GBM. Now, even with all the incredible advances in immunotherapy we've seen in recent years,
Right.
GBM still remains stubbornly resistant to treatment.
It's a tough one for sure.
It really is. So we need to find new ways to understand and target this disease.
Absolutely.
And that's where today's deep dive comes in. We're going to be exploring some cutting-edge research that's using a rather surprising model.
Surprising in a good way, I hope.
Oh, definitely. We're talking about dogs.
Oh, man's best friend coming to the rescue again.
Exactly. Canine high-grade gliomas, or HGGs.
HGGs, yep.
They actually mirror human GBMs in ways that traditional rodent models just can't.
You hit the nail on the head. Rodent models, while useful, they just don't capture the full complexity of the human immune system.
Right. They're really limited in terms of genetic and environmental heterogeneity.
Exactly. And that's where dogs come in. These tumors develop naturally in dogs in a complex immune system.
So it's a much more realistic view of what's happening in human patients.
Precisely. More translational, you could say.
Absolutely. So instead of lab mice, we're looking to our canine companions for clues.
And they have a lot to teach us.
Now, this deep dive is all about a specific challenge in both GBM and HGG treatment.
A challenge that's been a major roadblock in the field.
That's right. We're talking about the role of regulatory T cells, or TREGs.
TREGs, the guardians of immune balance.
Okay, so remind me, TREGs, they're like the immune system's peacekeepers, right?
Exactly. They prevent the immune system from going into overdrive and attacking healthy tissues.
So they're essential for preventing autoimmunity.
Precisely. But when it comes to cancer, this peacekeeping role can become a bit of a problem.
So how do these TREGs become a problem in the context of fighting cancer?
Well, in their effort to maintain balance, TREGs can actually suppress the immune system's ability to recognize and destroy tumor cells.
So it's like they're shielding the tumor from the very forces that could eliminate it?
So that's a great way to put it. They're inadvertently protecting the enemy.
That's the crux of the matter. And that's where this research paper we're diving into today comes in. The CCL2-CCR4 axis promotes regulatory T-cell trafficking to canine glioma tissues by Panek and colleagues.
A fascinating paper that really digs deep into the mechanisms behind TREG activity in canine HGG.
And as we mentioned, those findings have potential implications for human GBM as well.
Absolutely. Translational medicine at its finest.
Now, this paper was published in the Journal of Neuro-Oncology, which is a highly respected journal in the field.
Oh, definitely. A top-tier journal.
So let's get into the nitty-gritty. What exactly did they find?
Well, the key finding here is the identification of a specific signaling pathway.
Okay.
The CCL2-CCR4 axis.
CCL2-CCR4.
Yes, as a major driver of TREG recruitment to the tumor site.
So this pathway is essentially acting like a roadmap, guiding those TREGs right to the tumor.
Precisely. A sort of molecular homing beacon.
And here's where it gets really exciting. Disrupting this pathway could be the key to unleashing the immune system's full force against the tumor.
Now you're talking. That's the potential therapeutic implication here.
It's like removing the shield that's protecting the tumor, allowing the immune system to finally see it and attack it.
Exactly. A very promising avenue for new therapies.
This research could be a game changer for both our furry companions and human patients battling GBM.
I'm really excited about the potential here.
Me too. To understand how they got to this conclusion, let's delve into the research itself. They conducted a series of in vitro experiments, right?
That's correct. They worked with canine T cells and patient-derived glioma cell lines.
So they're working with cells outside of a living organism.
Yes. In a controlled laboratory setting, this allows for very precise manipulation and observation.
Makes sense. So their first step involved isolating TREGs from blood samples taken from healthy dogs.
Yes. They essentially extracted these peacekeeping cells.
But how did they make sure they were actually working with TREGs?
Good question. They confirmed the presence of FOXP3.
FOXP3?
Yes. It's a master regulator that defines TREG identity. Think of it as the TREG's ID card.
I see. So only true TREGs would have this FOXP3.
Exactly. And they also checked for CCR4.
Okay, and what is CCR4?
Well, it's the receptor that binds to CCL2. Remember that signaling pathway we talked about?
Right, so CCL2 is like a signal flare and CCR4 is the antenna that picks it up.
That's a great analogy.
Now, to study TREG movement, they used a Boyden chamber assay.
A classic technique for studying cell migration.
What exactly is a Boyden chamber assay, though?
Well, it's a clever setup that allows researchers to assess chemotaxis.
Chemotaxis?
Yes, which is the directed migration of cells in response to a chemical signal.
So it's like they're testing to see if the tregs are attracted to specific signal.
Exactly.
Hey.
In this case, they wanted to see if tregs would move towards CCL2 or substances produced by glioma cells.
So how does this Boyden chamber actually work?
Well, it's essentially a two-compartment chamber separated by a membrane.
Okay.
They introduced CCL2 and glioma cell supernatant, which is essentially the liquid medium the cells are growing in, into separate compartments of the Boyden chamber.
Got it. And
then they observed whether tregs migrated towards these compartments through the membrane.
so they essentially set up a choice for the tregs and observed their preference.
Precisely. A very elegant way to study chemotaxis.
Did they do anything else to manipulate the cells?
They did. They went a step further and used blocking agents.
Blocking agents.
Yes, to see if they could interrupt the CCL2-CCR4 interaction.
So they introduced roadblocks to see if they could disrupt the signaling pathway.
Precisely. A way to test the specificity of the interaction.
Clever. And what about the interaction between tregs and glioma cells?
For that they conducted co-culture experiments, growing both cell types together.
So they could see how the tregs and glioma cells influenced each other.
Exactly. It allowed them to observe how the presence of tregs affected glioma cell behavior.
It sounds like they really covered all the bases.
They were very thorough.
From isolating and confirming the tregs to testing their movement and manipulating their interactions with glioma cells.
We left no stone unturned.
Now for the big reveal. What did they find?
Their findings were quite insightful. And we'll dive into those right after this.
Sounds good. I'm on the edge of my seat.
Stay tuned. All right, so let's dig into those findings. Their findings really shed light on the intricate dance between tregs and glioma cells.
Okay, great.
First, they confirmed a direct relationship between CD25 expression and FOXP3 expression in canine T cells.
CD25, now that rings a bell. Remind me, CD25, that's like a volume knob on the treg's activity.
Yes, you got it. The higher the CD25 expression, the more active the treg.
Right, so it's like a measure of their peacekeeping power.
Exactly. It reinforces that these CD4 plus CD25 high cells are indeed bona fide tregs.
Got it. So they confirmed they were dealing with powerful peacekeepers.
Powerful indeed.
What else did they discover?
Well, they found that CCL2 acts like a powerful magnet drawing canine tregs towards the glioma cells.
So CCL2 is essentially guiding the tregs right to the tumor, like a homing beacon.
Precisely.
What happens when you block that signal?
Yeah. That's the burning question. And that's exactly what they tested when they used blocking agents to interfere with either CCL2 or CCR4.
So they're essentially throwing up roadblocks.
Exactly. And guess what? They observed a significant decrease in treg migration.
Wow. So they successfully disrupted the treg's journey to the tumor.
It seems so. Blocking either CCL2 or CCR4 significantly hampered their movement.
Fascinating. Were there any other findings that stood out?
There was one more crucial piece of the puzzle. They observed that the tregs themselves actually stimulated glioma cells to produce more CCL2.
Wait a minute, so the tumor is actually calling in reinforcements.
It's like a vicious cycle.
A self-perpetuating cycle of immunosuppression.
You got it.
This finding really highlights the need to break that cycle.
Absolutely. We need to disrupt that communication between the tumor and the tregs.
Now, you might be wondering, so what does this all mean for treating GBM and HGG?
An excellent question. And that's where things get really exciting because there are already therapeutic agents that target this pathway.
Really? Tell me more.
For instance, monoclonal antibodies can be designed to specifically target and neutralize either CCL2 or CCR4.
So we're talking about highly targeted therapies that can interfere with this specific signaling pathway.
Exactly. We're not just carpet bombing the whole immune system. We're going in with precision weapons.
Are there other approaches as well?
Yes. Small molecule inhibitors are another option.
Okay. And how do those work?
These molecules can bind to and block the interaction between CCL2 and CCR4, effectively disrupting the signal.
So it's like jamming the communication lines.
It's a good analogy.
It sounds like we have multiple tools in our arsenal to potentially disrupt this pathway. But how do we translate this research into actual treatments for patients?
That's where canine HGGs become even more valuable. They're not only a powerful model for understanding human GBM, but they also provide a unique opportunity to fast-track clinical trials.
I see what you mean. Since canine HGGs so closely resemble human GBMs, wouldn't it be much more efficient to test these treatments in dogs first?
Exactly. We can evaluate both the safety and effectiveness of these new therapies in dogs with HGG, and the data we gather could directly inform treatment strategies for humans.
It's a win-win situation. we can potentially help our furry friends while paving the way for groundbreaking treatments for humans.
Precisely.
It's a beautiful example of the power of translational medicine, bridging the gap between veterinary and human healthcare.
I completely agree. One health in action.
Now, while this research focused on the CCL2-CCR4 axis, it's important to remember that the tumor microenvironment is incredibly complex.
Oh, absolutely. It's like a bustling city with a million moving parts.
So you're saying there's a whole network of interactions happening, and this is just one piece of the puzzle.
Exactly. While this is a significant breakthrough in understanding one crucial pathway, there are likely other mechanisms involved in TREG recruitment that we still need to unravel.
It's like we've discovered a key route into the city, but there might be other secret passageways we haven't found yet.
A great analogy. We need to keep exploring, keep mapping out the terrain.
You're right. There were some important details about the methodology that are worth highlighting. For example, they used a very specific gating strategy with flow cytometry to isolate the TREGs based on their CD25 expression.
Yes, flow cytometry, a powerful tool for analyzing cells.
So they used a sophisticated sorting system to pinpoint the TREGs with the highest peacekeeping activity.
Exactly. They wanted to make sure they were working with the most potent TREGs.
Why is that detail so important?
Because it demonstrates a very precise and reliable method for identifying and isolating TREGs. It opens up a world of possibilities for future research.
It's like having a sharper tool to study these critical cells.
Precisely.
And this meticulous approach is reflected throughout their research. They even use specific canine glioma cell lines derived from actual patients.
Absolutely. Using patient-derived cell lines is critical because they provide a more authentic representation of the disease.
So these cell lines weren't just created in a lab, they came from real tumors.
Exactly. Making them a more accurate model for studying how the tumor environment influences TREG behavior.
That level of detail really adds another layer of credibility to their findings.
It definitely does.
Now, this research underscores the need for a balanced approach. Remember, the goal isn't necessarily to eliminate TREGs altogether.
Right. We need them for a healthy immune system.
It's about fine-tuning their activity, turning down their suppressive effects, specifically in the context of cancer.
You've hit the nail on the head. We need to find that sweet spot where the immune system is strong enough to fight the tumor, but not so strong that it attacks healthy tissues.
A delicate balance indeed.
It certainly is. But that's the beauty of immunology. It's all about maintaining harmony within this incredibly complex system.
Well said. And on that note, I think we've covered a lot of ground today.
We have.
This has been an incredible journey into a complex and rapidly evolving field of research.
It certainly has, and there's still so much more to discover.
Thank you for guiding us through it.
The pleasure was all mine. I always enjoy diving into these fascinating topics with you.
Me too. Now, I know our listeners are eager to hear your final thoughts and takeaways from this deep dive.
All right, well, let's move on to that then.
So we've uncovered some really promising findings in this research, but where do we go from here?
That's the million-dollar question, isn't it? We've identified this crucial pathway, the CCL2-CCR4 axis, that seems to be playing a key role in TREG recruitment to the tumor site.
Right.
But we can't just assume that blocking this pathway is a magic bullet.
Right. The tumor microenvironment is incredibly complex.
Exactly. It's a whole ecosystem of cells and signaling molecules all interacting in intricate ways.
So there's still a lot we don't know about how TREGs are being recruited and how they're interacting with the tumor.
Absolutely. This research gives us a really solid lead, but we need to keep digging, keep asking questions.
It sounds like every answer just leads to more fascinating questions.
That's the beauty of science, isn't it? It's a never-ending quest for knowledge.
Now, one thing that really struck me about this research is the power of translational medicine.
Yes, the bridge between veterinary and human healthcare.
We're learning from our canine companions and using those insights to advance human medicine.
Precisely. It's a win-win situation. We can potentially help our furry friends while also paving the way for groundbreaking treatments for humans.
It's a beautiful example of how a One Health approach can benefit both humans and animals.
I completely agree. We're all in this together.
So what's the next step in this research journey?
Well, I think one of the most exciting avenues is to start exploring those potential therapeutic agents we talked about, monoclonal antibodies, small molecule inhibitors.
Right, those targeted therapies that could disrupt the CCL2-CCR4 axis.
Exactly. We need to see if we can translate these promising findings from the lab into actual treatments for patients, both human and canine.
And those canine clinical trials could provide invaluable data that could accelerate the development of new therapies for human GBM.
Absolutely. It's a really exciting time to be in this field.
It certainly is. And this research really highlights the importance of collaboration between veterinary and human medicine.
Absolutely. We need to break down those silos and work together to tackle these complex health challenges.
Well said. This has been an incredible deep dive into a fascinating and rapidly evolving area of research.
It has, and I want to thank you for having me on to discuss it.
The pleasure was all mine. And I want to thank our listeners for joining us on this journey of discovery. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.